nutrient requirements by this different microbes, as we say, and these nutritional requirements, uh, uh, we have to limit ourselves to think of all the elements that are present in nature. There are only 12 natural environments that are needed for the microbial cell, and they are found in significant quantities. So we will classify them as either macro elements or macronutrients, and then the others will be, of course, the micronutrients, which are as trace elements. And in the macronutrients, you have a carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, potassium, calcium, magnesium, and iron. And then we also have some ions like sodium and chlorine, which are not meant to be a part of the as a nutrient, but they are definitely needed for the transfer of these nutrients across the membrane for you. So these macro elements are definitely needed in large quantities, relatively large quantities, whilst your micronutrients, such as your trace elements, which usually is provided by, usually in the water only, you don't have to give it in the nutrient media that you prepare. These are manganese, zinc, cobalt, molybdenum, nickel, and copper. These are definitely needed for certain enzyme for their functioning, and they are needed in very small amounts. So these are usually supplied in the water or in the media components. So we think about all organisms that require carbon, hydrogen, and oxygen, and of course they need an electron source also, where we will think of carbon being the backbone of all organic components present in the cell. Hydrogen and oxygen is always found in organic molecules also, so those are the major ones, carbon, hydrogen, and oxygen. And of course, your electrons, they are needed for energy production. They play a role in that. And they also are needed for reduction of carbon dioxide to form organic molecules. We'll study that later when we talk about photosynthesis also. Molecular oxygen, as we say that aerobic microbes require molecular oxygen, this is very seldom used in biosynthetic processes, even though it is required by the aerobic microorganisms, but it's not used in the biosynthetic processes. Some prokaryotes use oxygen as the electron acceptor, but some cannot grow in its presence. So oxygen being the criterion used to differentiate between aerobes and anaerobes. That's what we look at. So the requirements for carbon, hydrogen, and oxygen are usually satisfied together, and they will not only provide carbon sources that the microbes use, will not only provide hydrogen and oxygen, but also an electron source also. So we come across terms such as heterotrophic and autotrophic. So heterotrophic are those organisms that are considered to be heterotrophs that can use organic molecules as carbon source, and they also use the same carbon source as the energy source. So these are dependent on others to produce the organic molecules for them, while the autotrophs are the ones that we will find a lot of them now in our study here, that can use carbon dioxide as their sole or principal carbon source. And these are called autotrophs because they can build their own complex carbon compounds from carbon dioxide. So we will be studying a few of these autotrophs during our course here. When they use carbon dioxide as their only carbon source, they have to have another source for their energy. So they have to look for another compound that will be the source of energy for them. So based on the nutritional types, as we branch them off, based on their carbon source, we had them as heterotrophs and autotrophs. Now we have, based on their energy source, which they use, they are categorized as phototrophs and chemotrophs. Phototrophs, of course, use light as the energy source, whilst the chemotrophs use some chemical compounds and oxidation of these chemical compounds to derive energy for themselves. Then we have also the terminology based on electron sources. So lithotrophs are the ones that use reduced inorganic substances for their electron source, and organotrophs are the ones that obtain electrons from organic compounds. So you have 6 of those headings that we will put together now and we will say, do we have microbes in all these categories or not? Because up to now, what you have been looking at is the last category that is here, which are called chemoorganoheterotrophs. Chemoorganoheterotrophs are most of the microbes that you've studied, and all the life forms mainly belongs in this category, except of course in this microscopic world, we have different microbes now that we will be studying more so than these ones that you've been studying, right? So your chemoorganoheterotrophs use organic carbon for their carbon source. They use organic chemicals as their energy source, and the electron source is also organic. So this is the group that you have studied the most. We will now branch out and think about all these groups which use either carbon dioxide as their carbon source, light as their energy source, or inorganic electron sources also. So these will be the ones that we will talk about, are the sulfur oxidizing bacteria, hydrogen oxidizing bacteria, the methanogens, the nitrifying bacteria, iron oxiding bacteria. So these are the ones that we will focus on more so and compare them to the ones that you have already known as your everyday bacteria or fungi or your archaea or so. Okay. Besides having carbon, hydrogen, and oxygen as a requirement for microbes, of course, some others will be needed, elements such as your nitrogen, phosphorus, and sulfur. Of course, nitrogen, phosphorus, and sulfur become very important for amino acids and nucleic acids. So these molecules will have to be provided as nutrients for them. And nitrogen is, of course, supplied in numerous ways to the microbe. They can be supplied in the organic form, or they can be in the ammonia form, in the nitrate form also, where some organisms that we will study now that will be capable of assimilatory nitrate reduction. That means they will take nitrates and use this as a nitrogen source and assimilate the nitrogen into their other organic nitrogen compounds. So assimilatory nitrate reduction will be also a way to supply nitrogen to certain special groups of microbes that we will be studying now. And then a very, I would say, exclusive group is the ones that are nitrogen fixing microbes. They use the gaseous nitrogen, which is found in the atmosphere, which is very inert, and most living forms cannot use it. So to be capable of using this nitrogen gas and be able to fix that inert nitrogen into nitrogenous compounds, we are going to be looking at some of those. And the process that is involved in the nitrogen fixation using different bacteria will be definitely looked at in this later in this course now. Phosphorus usually supplied as inorganic phosphate for most living forms. Sulfur also is supplied as sulfate, where it uses the sulfate in the assimilatory sulfate reduction, where they will assimilate the sulfur from the sulfate and put it into organic compounds for the microbe. Besides these carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus, there are certain growth factors that are needed for the growth of some microbes. These are usually organic compounds, and they are essential as precursors to the cell components that we will look at. The cell cannot synthesize them, and so they have to be provided in their nutrients for them. And these growth factors, of course, will be needed for their survival and reproduction. And there are different classes of growth factors that we look at. Some microbes will require amino acids as growth factors. They are not able to synthesize those amino acids, and those amino acids have to be provided in their nutrients. And of course, this amino acids will be used for protein synthesis, while some of them will require purines and pyrimidines, which will be needed for your nucleic acid synthesis, such as your DNA and RNA. And vitamins may be needed by some as their growth factors, and they function as enzyme cofactors. And we'll look at quite a few of these cofactors. There we will be dealing with a lot of biochemistry here, thinking about enzyme cofactors and how they drive your reactions. So here are some examples of some common vitamins. that are needed in different functions in the microbe. For example, we will be talking about carboxylation or carbon dioxide fixation, the top of the list here. These are the ones that are capable of carboxylation or carbon dioxide fixation. And this is usually seen in one carbon metabolism. This one carbon metabolism is very much dependent on the vitamin biotin. So this biotin has to be supplied for the growth of such bacteria, such as your Saccharomyces cerevisiae and Leuconostoc. The B, F, and A, as you know, comes from which group they belong to. B is for the bacteria, F is, of course, for the fungi, and A is for the archaea. So we're giving you examples of different bacteria or, protozoa, P is for the protozoa, different groups of microorganisms that require vitamins for their different metabolic processes. So one carbon metabolism definitely you will see being repeated again and again, where different types of vitamins are needed. So we'll talk about folic acid a little bit, this B12 vitamin, biotin, niacin, riboflavin, thiamine. We will come across all this information as we look at the pathways that these very novel microbes utilize for one carbon metabolism, or when we go to even think about your pyruvate decarboxylation, the common one that you looked at, transamination reactions and all of them will require certain vitamins as the cofactors in the enzymes. So let's come to really the structures and try to look at some of the structures for today. We will not go through all of them, but I'm going to emphasize some of the external structures that may be found on bacteria. And of course, the two types of external appendages that we find on bacteria are categorized either as flagella or fimbriae. Those are the two names we come across. And this is, of course, showing your Salmonella typhimurium, which is showing the flagella and the fimbriae. The flagella are, of course, much thicker and larger, and your fimbriae are finer and smaller. So what are some of the functions of these external structures that we will look at? They, of course, are needed for attachment to surfaces, or in some cases, they will be for horizontal gene transfer, such as conjugation and so on. And you will see these structures also needed for cell movement to be able to move. The terms that you will come across are pili and fimbriae and flagella. Those are the names that will be used for these. So if you look at pili and fimbriae first, these are now the authors differ on this. We used to call them fimbriae and sexpili or pili, but we have just added the prefix of sexpili, where you want to tell, where you want to differentiate a pilus that is capable of allowing some conjugation to occur. But the word just pilus or pili can be used interchangeably with the fimbriae. But if you're talking about the sex pilus, then it is definitely similar to the fimbriae, but they're longer and thicker, and they're less numerous, smaller in number, and they are capable of allowing the F pilus mediated conjugation. So you're familiar with the idea of bacteria capable of conjugation in nature. So this is what this pilus, F pilus, or the sex pilus will be used just for conjugation. Whilst the regular pili and the fimbriae, they may be short or thin, hair-like. They are proteinaceous. That means they are composed of proteins, and they mediate attachment to surfaces. There is a special type of fimbriae, which we will discuss later, type 4 fimbriae, which is required not only for motility, but also to be able to take up the DNA. So the DNA uptake, when these horizontal gene transfers will occur, will require a synthesis of a special type of fimbriae called the type 4 fimbriae. Of course, the F pilus genes are found on the plasmid. So this is a plasmid property found on this extrachromosomal DNA, which will give the property of producing this F pilus, which will allow the cell to act as a donor. Flagella are, of course, locomotor appendages. We call them locomotor because we know that cells that have flagella are capable of a swimming motility. They also are thread-like, and they function in motility and swarming behavior also. In some cases, the flagella may also be as a means to attach to surfaces, and we also know that they may be labeled as virulence factors. So let's talk about virulence factors. Are you aware of what a virulence factor is? Anybody? The word virulence is, of course, the measure of pathogenicity. A bacterium may be pathogenic, but it becomes more virulent if it has some of these virulence factors. For example, a capsule production is considered to be a virulence factor. In some cases, the flagella may also be considered a virulence factor, meaning a pathogenic bacterium may become more virulent if it has these special flagella. Now, when you talk about E. coli 0157H7, are you familiar with that E. coli? What is so special about E. coli 0157H7?
Does it cause diarrhea?
It is a pathogenic form which causes hemolytic uremic syndrome. HUS is what I call it. HUS is the hemolytic uremic syndrome, where there will be blood in the urine. That means it is affecting the kidneys. It is not the one that causes diarrhea one. This is the one that causes a much severe disease. And this 0157H7 was categorized to be different from your regular E. coli. Your regular E. coli is present in everybody's colon and is considered to be non-pathogenic, whilst the E. coli 0157H7 has been categorized as a pathogen. Also, not only it is normally not found in humans, it is found in certain cows. It is a resident of certain cows in their gut, and it is the one that will cause a severe disease in humans. So 0157H7, if you look at it, when it was categorized as O and the H, those are two antigens that are present on this E. coli. And O is the somatic or the cell wall antigen, whilst H is the flagellar antigen. So that means the antigen present on this specific E. coli is very different from the flagellar antigen that is found on your normal E. coli. So this categorization of H antigen is always related to a flagella. Okay, so if you ever see H antigen being talked about in any bacterial cell, you know it is a flagellar antigen. And if it is an O antigen, then it is a cell surface antigen, which we call the somatic antigen. So definitely telling you that virulence factor, that flagella, certain flagella will act as a virulence factor, and we can categorize some microbes based on their H antigen to have become more virulent than the normal kind. Does this make sense to you now?
Yes, Dr. Rahman, it all makes sense. It's a nice review.
Good. So let's look at the structure of the flagella. Of course, it's a thin, rigid protein structure. We cannot see it under the bright field microscopy because they are so small, about 20 nanometer diameter. You have to do special staining so that you can coat it to make it thicker, to make it visible if you're trying to see them under your light microscope. But of course, you can see it in your electron microscope pretty clearly. And when we look at it, we see three ultrastructures, the three parts that are found on the flagella. And we will look at the structure in the next slide. But the patterns of flagellation has also been described. So you should be familiar with the terms called monotrichous, amphitrichous, lophotrichous, and peritrichous. So monotrichous has one flagellum. Polar flagellum will be called the one where the flagellum is at the end of the cell. Amphitrichous is the one that flagellum is on both the sides. Amphi, again, meaning both sides. Lophotrichous, a cluster on one or both sides coming out as a cluster. And peritrichous, where the flagella are arranged on the entire surface. Now, to tell you that if you're familiar with this idea that we have up to now, most of the bacteria that have flagella, they are supposed to be in a rod shape. Even if they are a short rod, cocobacillary form also, they can have flagella, but it is not seen to have that spherical bacteria having flagella. So it seems to be some properties within the form of the bacteria that allows them to make a flagella, because this flagella is supposed to protrude out of the cell wall, and the cell wall structure becomes very different and important in the cocci versus the rod shape. And we'll look at that very soon as to how this arrangement would occur. So as such, a bacterium, when it is monotrichous, this is a genetic property that it will always be monotrichous, and some of them will be amphitrichous. So we can use this property for differentiation and characterization of different bacteria. So here is the flagellar distribution kind of a picture for you, where this is Pseudomonas. You're familiar with Pseudomonas. It is monotrichous, and it has a polar flagellation. So there's one pole, one flagellum at the pole. We know Pseudomonas is very, very motile, and yet it has only one flagellum, which propels it into a liquid space very, very fast. Spirillum are the bacteria that have lophotrichous flagellation. There they have a bunch of flagella coming from one end of the rod here. And here is your totally motile bacterium called Proteus vulgaris. You're familiar with it. This Proteus vulgaris has peritrichous flagellation, and you can see the flagella are so long. Of course, this is a chain of cells. This is not one cell. This is a chain of cells where all the flagella have been stained with a special procedure where you can see them like hair-like structures, long enough, and this makes them very motile, very fast swimmers, as I call them. So coming to the parts of the flagella.